Okay, so let's finish up our little lecture here on meiosis. So as we said in the last part, meiosis is a process in which you make a gamete, which is a sex cell. It's very different from mitosis. In mitosis, you're making a somatic cell. So let's go through the differences between these two processes, and then we'll finish up by talking about something called a karyotype. Okay, so if you take a look up here in the corner, let me just change this a little bit. There we go. You see this little diagram. I know it's small, but I'll be referring to it a little bit, but it's more about what's written over here. Okay, so first off, where do these processes occur? Well, in mitosis, this is going to be for your body or somatic cells, and in meiosis, this is only going to be for your gametes. So, do asexual organisms go through meiosis? The answer to that question would be no, they do not, right? They don't need to, because a single cell becomes two new cells. It's just mitosis. It's only in organisms that reproduce sexually that meiosis occurs, and it occurs in their gametes. The next big difference between these two processes is the number of divisions. In mitosis, there is a single division. One cell splits into two. You get one diploid cell splitting into two new genetically identical diploid cells, where in meiosis, there are two divisions. So if you look at this diagram here, this shows mitosis, prophase, metaphase, anaphase, and then telophase. You end up with two new cells that are genetically identical to the original cell. In meiosis, however, you get your first division, and where you're going to get two cells, which are going to be haploid, and then that's going to divide again, and you're going to get four new cells, which are going to be half of the number of the original cells. So one division, two divisions. And that leads us to the next difference, which I pretty much just said, but in mitosis, one diploid cell becomes two new diploid cells, and in meiosis, one diploid cell becomes four haploid cells. And that's the big difference there, the number of cells that come out and their ploidy, the amount of chromosomes in there. The next big difference is genetic variation. In mitosis, there is none. It's just a single cell becomes two new genetically identical cells. In meiosis, however, because of crossing over, when those tetrads form, there's that shuffling of the deck that occurs every single time meiosis occurs, and that gets you genetic variation, which allows for organisms to survive under different conditions. And we'll talk more about that when we get to evolution, but what this allows for is cases like, for example, the coronavirus, where some people with immune systems can fight it off and some other people's immune systems cannot, and the people that can fight it off are going to survive, and they're going to pass on those traits. That is how evolution occurs, and genetic variation is pivotal to that. Okay, and lastly, we're going to have this thing called a karyotype that we're going to talk about. Now, as we said before, there are genetic conditions that can occur, genetic mutations, when a person, is making a new organism, going through this process. Sometimes things happen. So we use a process called karyotyping in order to determine if somebody has a genetic abnormality or if they have a normal genetic makeup. So what is a karyotype? Well, really all it is it's a tool. It's a way to see if somebody has a genetic mutation. And what they do is it's a pretty simple process. They have a cell, and when a cell is in the process of metaphase, when it's in the metaphase stage, when all those chromosomes have separated out and lined up, they take a picture of it. And then they use computers to really just separate them out and pair them up into their chromatids. So if you take a look here, you can see this would be an example of a karyotype. Why do we use it? We use it to diagnose genetic abnormality. So for example, you could see if somebody has trisomy 21 if you're doing this, which is Down syndrome. So what you're going to see here is you're going to see 46 chromosomes. Each are going to be broken into their pairs, or 23 pairs. So we have 46 individual chromatids, which are going to be paired up into their pairs of 23 pairs, right? So this would be the first set, second, third, fourth, fifth, all the way down to the 22nd here, and then our 23rd pair over here. So each of these individual chromatids is paired up with its identical chromatid. Now, remember, where do these chromatids come from? Mom, dad, mom, dad, mom, dad. It's one from each parent. So the first 22 chromatids, or chromosomes here, are going to be called autosomes, and they're identical in males and females. So if I was looking at this, I can tell you that this is a male, and we'll talk about how I know that in a minute. But for numbers 1 through 22, I wouldn't be able to tell that, because in males and females that are identical, they're the same. Males and females have the same numbers 1 through 22. It is only this 23rd pair here, which we call the sex chromosomes. This is what's different in males and females. So a female has two X's and a male has an X and a Y. So looking at this karyotype right here, I can tell you it's a male. And the reason I can tell you it's a male is because this individual has an X chromosome and a Y chromosome. Now, the other 22 are going to be the same. I wouldn't be able to tell from that. So if I just showed you this without this last pair right here, you couldn't tell if it was a male or a female. You would need to know this last pair, which is the 23rd pair. So let's take a look at some examples and see if we can figure this out. So this is an actual karyotype. So if you take a look here, you can see these chromosomes have been paired up into their, let me move this down a little bit. Oops, I just gave you the answer. Well, you don't know what Turner syndrome is, so that's fine. So if you take a look here and you look at this set of chromosomes, let's take a look at it. You can see there are 23 pairs, numbers 1 through 22, and then the 23rd pair. So let's take a moment here and look at it and see if you can figure out what's wrong with this. This is an abnormal karyotype. So take a minute and look at it and see if you can figure out what's wrong with it. So look at all the chromosomes, look at how they're paired up, and see if something looks weird. Okay, so hopefully you picked up on this, but what you hopefully noticed is that there is no 23rd chromosome. There's a missing one. So there's just one chromosome. There's no X or a Y. So this is called Turner syndrome. So you saw that answer flash up. But if you don't know what Turner syndrome is a condition where a female has only one X chromosome. You may be saying to yourself, wait a second, Mr. Adolf, how do you know if it's a female if there's no Y here? Well, what makes somebody a male is very simply if they have a Y or not. If they don't have a Y chromosome, they do not have the ability, they're not a male. It's as simple as that. So this is a female with one X chromosome. And Turner syndrome is a disorder in which a female has only one X. And we talked a little bit about this one in class, actually. You would have a situation where a female is born with one X and they're going to have more male type characteristics. So a female with Turner syndrome, they're going to have broader shoulders, more body hair. They're just going to have more male type characteristics, traditionally male physical characteristics. Let's look at the next one. So if we take a look at this one, there's something wrong with this one. And so everyone take a look at it and see if you can figure out what's wrong with this karyotype. Okay, so hopefully you had a chance to take a look here. If you take a look at numbers 1 through 22, and then the 23rd chromosome here are the sex chromosomes. So let's take a look at this together. First off, is this a male or a female? Well, we know it's a female. And the reason why is because it has two X chromosomes. There's no Y chromosome, so it's two X chromosomes. There's nothing wrong with those, so we know it's a normal female in terms of sex chromosomes, but there's something wrong with it. So if you take a look here, you can see, hopefully you notice that on the 21st pair, instead of having only two chromosomes, we have three. So this person has trisomy 21. So this would be a karyotype of somebody that has Down syndrome. Everything is normal except for this 21st pair, which is going to be called trisomy 21. And as we talked about in class, we talked about the physical characteristics of somebody with Down syndrome. We'll try one more. Okay, let's take a look at this last one and see if you can figure out what's wrong with this one. And I will be quiet for a moment and let you take a look at it. Okay, I'm really bad at wait time. I'm bad at that in the classroom, and I guess I'm bad at it here too. I don't like, even if I'm the one talking, I don't like silence. So let's see if you guys can figure this out. So if you take a look here, first of all, is this a male or a female? Well, this is a male, and the way I know it's a male is because there's an X and a Y. So if there was two X's and no Y, I would know it's a female, but there's an X and a Y here, and that is going to be a male. Now, this is not a normal male though, because if you haven't noticed, if you take a look at the other autosomes here, if you look at number 18, there are 3 chromosomes there, and that is called Edwards syndrome. And in Edwards syndrome, that is a genetic disorder where somebody has an extra 18th chromosome, and it leads to a bunch of physical and emotional abnormalities. So that is a karyotype. So we're going to have an activity where you're going to have to try to do some work with these a little later on, but that's it. Thank you, guys.